Thanks for coming to today's seminar. For those of you who don't know me, my name is Derek Sowers. I'm going to introduce today's guest speaker. I'm both a CCOM graduate student right now and also a co-worker of our guest speaker, Phil Trowbridge. We both work currently for the Piscataqua Region Estuaries Partnership, which we'll refer to through the rest of the talk as PREP, to keep things simple. It's one of 28 national estuary programs that are funded by EPA. The purpose is to monitor, restore, and protect nationally significant estuaries. Here in New Hampshire, we have Great Bay and Hampton-Seabrook Harbor, which is the focus of today's talk. I just wanted to let you folks know that PREP has been a part of UNH Marine School actually since 2005, and so we're part of the new School of Marine Science and Ocean Engineering. And we thought today would be a good time to just kind of make you more aware of what that program's all about, the kind of data synthesis that happens there, and talk about ways we might be able to collaborate more closely with the resources here at CCOM. So a little bit of background on Phil. He's the coastal scientist for PREP. He works on managing our research and monitoring programs, synthesizing a huge amount of data for the region and the region's watershed. In addition to his role at PREP, he is the Water Quality Standards Program Manager at New Hampshire Department of Environmental Services. He has a Master's of Science in Civil Engineering, Civil and Environmental Engineering from MIT, and he's a licensed professional engineer. This past year, Phil was named Conservationist of the Year by the Coastal Conservation Association of New Hampshire. So, also, I wanted to thank Phil for coming to give this talk on short notice. He's flying out for a 2-week trip to Guatemala for a language immersion program, so thanks for coming.
Thank you. And thank you all for being here. I was driving over from Concord thinking what a dreary Friday afternoon it is. And I'm sure with Thanksgiving right around the corner, everyone's got lots of things to do, so I'm really glad to see so many people here. As Derek mentioned, I am leaving tomorrow at 11 a.m. for a 2-week Spanish language immersion class in Guatemala. So when I agreed to give this talk, I first thought, maybe I should just do this all in Spanish, get a kickstart on it. But I really didn't think I could do the subject justice. Although all morning I've been torturing my coworkers by only responding to their questions in Spanish. And so when you're trying to leave on a 2-week vacation and people asking you where the files are and what you should do and this or that, and you only reply in Spanish, it hasn't been a particularly smooth handoff. But it's been fun for me nonetheless. All right, I'd like to do 3 things today. First, I want to introduce you to PREP, for those of you who don't know about us. So who we are, what we do, and our connection to the Marine program. Then I want to run through some of the findings we have from our State of the Estuaries report. And then I want to end by talking about some of our current research that we're working on and how we can partner with CCOM on that research. Okay, so to start with, one of the things you need to know about PREP is that we're part of the National Estuary Program. The National Estuary Program was created by the Clean Water Act, and it's administered by EPA. There's 28 programs around the country, and we are one of those 28. We are in illustrious company: Puget Sound, San Francisco Bay, Long Island Sound, Charlotte Harbor, and right in our backyard, a national treasure, the Great Bay Estuary and Hampton-Seabrook Harbor. Another thing to know about us is our study area. This is a map of the watersheds draining to the Great Bay Estuary and Hampton-Seabrook Harbor. This covers over 1,000 square miles. There's 52 towns that are majority in the watershed, and that's split by the state line between New Hampshire and Maine. So an important thing to know about us is that we're a multi-state program. And that's a big difference from some of the other organizations that do environmental conservation, because those are often state-based programs, and they end at the state line. So for instance, my other job at the Department of Environmental Services, I have a hard time crossing the state line into Maine. And I likewise have colleagues from Maine who have a hard time getting to meetings in New Hampshire. So it's nice to have an organization that works across state lines and works regionally. Third thing I'd like you to know is that we have a plan. There's been a lot of talk about Great Bay and the estuary lately, and there's a lot of casting about for what's the plan? How are we going to fix the problem? What are we going to do to preserve the estuary that we all care for? Well, PREP's been ahead of this from the start. We worked on a 10-year plan using 120 different stakeholders for a restoration of the Great Bay Estuary in Hampton-Seabrook Harbor. It's a very detailed plan. It has hundreds of action items. Really all we need to do is dig down and implement the plan. Another thing to know about us, we're stakeholder driven. This was written into the Clean Water Act, Section 320, that created the National Estuarine Program. This is a bottom-up type program. These logos just represent some of the partners we work with for monitoring. These are also people that sit on our management committee, which is like a board of directors. We also have a science and technical advisory committee that's made up of researchers from largely University of New Hampshire, but also from New Hampshire and Maine and federal agencies. And finally, we handle a lot of data. Every 3 years, we do a State of the Estuaries report. The State of the Estuaries report covers a wide range of topics, from land use, water quality, habitats, species, beach closures. And there's a lot of other things that we collect as ancillary information to help us interpret those as well. We estimated at the start of this process that we handled well over 1 million individual data points. And I'm sure for the CCOM people here, that's not a lot. You deal in millions and billions of pixels and stuff, but we're talking individual grab samples of water, and people out collecting fish and fish ladders. That's a lot of points collected the hard way. Not to say that your data is not collected the hard way. And then we go through a distillation process where we're trying to summarize that data into tables and graphs. And then we're trying to convert those into indicators, which is what we use for our report. And then eventually we get it down to a report. But through that process, we come to manage a lot of data. We come to have a lot of data. And one of our reasons for being here today is to make sure you're aware of this data. And the PREP is part of the Marine School. We were looking for ways to partner with other programs within the Marine School. We wanted to make sure you knew that we had this information. So if it's of interest to you for your research, please let us know and we can share it so that you don't have to go through the painstaking process that we already have. Okay, so now I'm going to talk from our report, the State of the Estuaries report, and just by means of introduction, we use indicators in our report. And this may not be new to you, but it is new to some people. So I want to introduce or define what an indicator is. An indicator we use is a statistic or metric that summarizes data for some condition or trend in the estuary. And we summarize our indicators into 3 groupings: pressure indicators, condition indicators, and response indicators. And as you can guess, the pressure indicators relate to human pressures on the estuary. Condition indicators represent some sort of status or trend within the estuary. And most of our data falls into this category, because this is more like what traditional monitoring programs develop. And then response indicators are measurements of how we are responding to the threats on the estuary. So things like how we're protecting land, how we're restoring habitats, how we are responding to the concerns. Now, the report is 22 indicators long. That's not every possible threat, that's not every possible condition, and that's not every possible response. But they are a very wide range of indicators, and they cover a large range of topics. And we feel that these, as a result, they do, that the overall report is representative of the state of our estuaries. So we'll start now with some of our pressure indicators. The impervious cover, just to define it, is things like roadways, roofs, parking lots that water can't pass through. Precipitation onto these surfaces runs off rapidly, forming stormwater, and affects the water quality in our streams, in our lakes, in our estuaries. We know from research elsewhere that if you keep the percent of imperviousness low, that's a way to improve water quality in your watershed. And so we've been tracking this data for the whole watershed, this 1,000 square mile watershed, since 1990. So as you can see, the pattern has been a pretty steady increase of impervious surfaces over this time. On average, we're adding 1,800 acres of impervious surfaces to this watershed every year. And over this time, we have grown to the point where we are nearly reaching a 10% imperviousness for the entire watershed.
So how do you collect that data? Is that aerial photography or? Yeah,
this is with Landsat TM, and it's processed by Faye Rubin's group at Complex Systems. And we're doing an algorithm that's a sub-pixel analysis. We are thinking about, and we actually want to head towards a more detailed analysis for the next round, but from a ability to cover, this is the best that we could do in 1990, and we wanted to continue with the same method as long as possible. But we're recognizing that probably for the next report, we're going to switch to a new method. So to put this growth in perspective, since 1990, there's been a 120% increase of impervious surfaces in our watershed. And during that same time, the population of the watershed has increased by 19%. So clearly a more rapid growth of imperviousness than population growth. Well, there was a picture here, but it's gone. So we won't dwell on that. But we also look at this information spatially, and you can see a pattern of watersheds that have more imperviousness closer to the coast than further away. All right, nutrients and nitrogen. It's not a topic that anyone has any interest in, right? It's not that important. Anyway, nitrogen is an important nutrient in the estuary. However, we know that if you have too much of a good thing, you can have problems such as excessive algae growth, which then leads to problems like loss of underwater species and low dissolved oxygen. So we've been tracking the load of nitrogen that comes into the estuary since 2003. And this is through monitoring that we do at all the different tidal dams that flow into the estuary. We split this load up into two pieces. We have the blue bars is the watershed load, which is load from diffuse non-point sources such as fertilizer, septic systems, atmospheric deposition. And the red bars here are the loads from the wastewater treatment plants. And just for reference, we put this black line on here showing the annual precipitation. You can see that there is clearly a pattern with precipitation in terms of the load of nitrogen coming into the estuary. During our highest rainfall years, we had a spike up of the non-point source load. The load from the wastewater treatment plants has remained relatively stable during that period. Overall, when you just want to look at the total load coming in and where the sources are coming from, about a third of it from a total nitrogen on which is the graph on the left-hand side, is coming from wastewater treatment plants, and 2/3 of it coming from the watershed sources. That's for total nitrogen. Now there's many forms of nitrogen. The form that is of most concern for algae growth is dissolved inorganic nitrogen, and that's shown here in the pie chart on the right. Again, splitting out the loads in terms of whether it comes from a point source or a non-point source. DIN is about 52% provided by wastewater treatment plants and 48% provided by the non-point sources. So related to nutrient loads, obviously, is nutrient concentrations. We've been monitoring dissolved inorganic nitrogen in the middle of the Great Bay since the 70s. And I say we as in UNH. And let me explain how this trend graph, what's going on with this trend graph, because we will be looking at a few of these as we go through here. We have year on the x-axis, concentration on the y-axis. And these blue dots are the annual averages for years where there's complete data. We're monitoring this monthly, and not all years did all the monthly samples get collected. These squares represent annual averages for years where there was incomplete data. So we wanted to represent that data, but show it in a different way. The error bars are the standard deviation of the average, standard deviation of the concentrations observed during the year. And then we've put this dashed line in here to indicate the last 3 years. So when you look across this whole data set, the concentrations of dissolved inorganic nitrogen have been increasing since 1970. You can see this pattern here of much lower concentrations in the late 70s, with a spike up in the ramping up to 2000, and then some variable concentrations since then. It's the same information, but for chlorophyll. So our measurements of chlorophyll as an indicator of microscopic algae. It's a lot of variability in algae, and monthly sampling is not particularly useful at characterizing all of that variability. And over this full data set, we're not seeing a significant trend. But one of the things that we've discovered is that microscopic algae is not the only real algae that's important in the Great Bay. There's also macroalgae, things like ulva, which is shown in this picture here. In the shallow areas of Great Bay, those tend to be a bit more of a concern. We don't have nearly the data on macroalgae that we have on chlorophyll, because it's not something that's easily measured. You can't go out and collect a bottle of water and then look for macroalgae. It's much more of a habitat mapping program. But we do have 2 studies that were done many years apart that use the same methods. So believe it or not, this is a point. There's 2 points on this graph. And I know none of you are going to be happy about the graph with 2 points. But there's way more points in here than the 2 points. These are the averages of 2 years. Each study is a 2-year study that involved about 100 measurements each. And this is the average and standard deviation for each of those. So in this location, which is in the middle, on the shores of Great Bay near Lumberland Creek, we saw an increase in the proliferation of ulva from about less than 1% in the 80s up to 39%. This matches the anecdotal observations that have been reported to us by the eelgrass mappers and the other benthic biologists in Jackson Lab of a large amount of ulva and macroalgae forming green tides in the intertidal areas of the bay. So for dissolved oxygen, we're fortunate in the estuary to have a nice network of buoys that monitor this parameter every 15 minutes. There's actually 6 of them, or 7 if you count the Great Bay Coastal Buoy. There's 1 in the middle of Great Bay, and then there are 3 in the rivers and 1 at the mouth of Portsmouth Harbor. For dissolved oxygen, there's a state standard for what is the acceptable amount of oxygen. It's essentially 5 milligrams per liter instantaneously, or 75% saturation on a daily average. And what we do for our indicator is just chart the number of days where we have violations of those standards. Now, the way these charts work is that for each year, we've categorized the number of days in terms of whether they're in compliance with the standard, which is green, or there's no data, which is gray, or if there's a violation of the standard, it's red. So what these 2 charts summarize the overall message about dissolved oxygen. In places like Great Bay and Portsmouth Harbor, our large bays and harbors, we generally are not seeing problems with dissolved oxygen. We have compliance with our standard most of the time. But in places like the Lamprey River, the Oyster River, the Squamscott River, and the Salmon Falls River, where we monitor this, we see consistent violations of the dissolved oxygen standard. And sometimes those periods of violation last up to two or 3 weeks during the summer period. So eelgrass. Eelgrass is an underwater habitat. It's the base of the benthic food chain. It stabilizes sediments, it filters water, and provides important sanctuary for shipfish and shellfish. Now, eelgrass is important not just for its own case, but also because it is indicative of the health of the estuary. It's important for the survival of other species as well. In Great Bay, we're lucky to have one of the longest records of daily eelgrass mapping, or sorry, of yearly eelgrass mapping anywhere in the world. Fred Shore's been mapping this eelgrass since 1981, and we have data through 2012. There's an interesting pattern of the eelgrass. First, oh, geez, sorry, I keep hitting the wrong buttons. First, you'll notice this period here in the late 80s. There's a disease that affects eelgrass called wasting disease. And there was an outbreak of that in the late 80s, and you can see a large dieback of the population, but then a rapid rebound afterwards. Then, since about 1996 or so, the pattern has been a slow decline, not attributable to the wasting disease. Current populations of eelgrass are down about 35% from what they were in 1996. And there's been, while there has been variability in these last few years, the overall trend is still significantly downward. Suspended sediments. Now, I have to apologize for this graph. This is the case where the graphic design took over. It didn't, this graph had to be smushed a bit to fit within a part of our report, so it really doesn't look like much. But if you expanded the axis here, you would see the trend. We've been monitoring suspended sediments in the estuary for this same period back to the 70s. Suspended sediments is related to loads into the bay from the watersheds, as well as resuspension within the bay due to wave action and other patterns. And during this period of time, we've seen a significant increase of suspended sediments, which can affect both the underwater habitats of eelgrass, where it needs light to be able to survive. It can also smother oyster beds and impact the fronds of the eelgrass eel leaves as well. These 3 slides here are talking about bacteria. Bacteria is important in our estuary for 3 reasons. Well, one is our beach closures. Beaches are an incredibly important tourist issue for us here in the coast. And when beaches are closed, that's most often due to bacteria. And another has to do with shellfish harvesting. And then finally, it's an indication of illicit connections or other pollution sources. These 3 graphs are showing a little bit different things. In the upper right, it's just the concentrations of fecal coliform bacteria in the estuary during dry weather. If you were to look at this with wet weather, it's extremely variable, and it's not very conducive to looking at trends. But in dry weather, we're seeing a long-term decline in the bacteria concentrations. In some cases, down 50 to 92%, depending on which station you're looking at. This bottom right figure deals with beach closures. The little blue line deals with the number of beach advisories that have been issued, but the orange bars are the percent of the time that these beaches are under advisory. So as you can see, there have been some issues. We've issued beach advisories. We've had to close beaches due to bacteria, but on average, the percent of the time that we're closed is very low. It's about 1% or less. And that's a very low number compared to other states. And New Hampshire actually is always very close. We're either number 1 or number 2 in the country in terms of our health of our beaches, as we're ranked by NRDC each year. We're in this rivalry with Delaware, and they cheat, so they get number 1. And we, we really should be number 1. They fudge their numbers somehow. And finally, the pie chart deals with shellfish harvesting. If you want to harvest shellfish and eat them, you have to take it from an approved area. FDA has pretty strict requirements of what the water quality is to be in that approved area. It's as if you were going to eat the shellfish raw. So it has to have low bacteria concentrations. For us, about 36%, only 36% of our waters are open year-round. And then the rest is closed all the time. And even within this 36% that are open, we only can open if there's a rainfall event that causes bacteria problems, we have to close it. And so those are closed another 42% of the time. So when you think of all the opportunities that are lost here of harvesting, it's a pretty small amount, or we actually have a very small amount of all the potential harvesting opportunities that we could have. So it's a mixed message with bacteria. We see declining trends. We generally are not seeing problems with beach closures, but we are certainly not open to the fullest extent we can for shellfish harvesting. So our indicator for measuring toxic contaminants is the concentrations of these contaminants in the flesh of blue mussels. It's very expensive. Sorry, yeah, go ahead.
Sorry, are those shellfish closures though, are those due primarily to the harmful algal blooms, or does that play a role in that at all?
It does play a role. It's a smaller role. For the most part, the closures are due to local sources of just bacteria. But when we do have an offshore bloom that comes in and causes us to shut down for red tide, that would be part of these closures. Those don't normally proceed much past Dover Point, and a large portion of our waters are inshore of that. Yeah. Blue mussels, a very prolific organism, also a filter feeder, and an excellent indicator of the water quality for toxic contaminants. Really difficult to test for toxic contaminants in the water because the concentrations are so low. But if you collect blue mussels and process their tissue, you get these great patterns of the changes of the water quality that they're filtering. We have 3 areas that we typically monitor. One is in Hampton Harbor, one is in Portsmouth Harbor, and one is at Dover Point. We have declining trends in all of these areas for legacy pollutants. For instance, this graph here shows the change in PCBs in Portsmouth Harbor. And this is a site on the Navy shipyard, which is one of the more polluted areas in the region. And this is, every year, for me to get this data is a huge security nightmare. When we decided to do this site as our long-term trend site, we did not have any idea how hard it was going to be to get on a Navy base every year with equipment and vehicles and et cetera. This pattern here is for DDT, the pesticide in Hampton Harbor, and this trend here is showing lead in mussel tissue at Dover Point. All of these, again, are legacy pollutants. Steps were taken years ago to remove these pollutants from the waste stream. And what this is demonstrating is now it's still paying dividends, that this type of management action is important to take. Another shellfish that we look at is oysters. And in this case, we're looking at just the abundance of oysters in the bay. Oysters are an important filter feeder. They form reefs, which is important habitat for other species. They filter water, they clean water, and they stabilize sediments. Overall, the pattern, though, of oysters in our bay has been a declining one. We started with over 25 million oysters in 1993, and that's dropped down to a low point of 2000 of about 1 million. Since then, we've had a slow climb up to about 2 million, or 2.2 million, which is 22% of our goal for restoration. But that's a really small number compared to what it used to be. We spent a little time researching some of the historical accounts of this area, and oysters were so plentiful in the early colonial days that they were used to feed the livestock. So we've fallen quite a ways down from there, but the overall pattern has been decline. The issue that we're fighting mainly on this is diseases, MSX and dermo. These were invasive species that came into Chesapeake Bay and then have been doing range expansions further north and eventually hit us in the late 1990s. The other recreationally important shellfish is clams, and while Great Bay is the important place for oysters, Hampton Harbor is the important place for clams. We've been, the clam populations have been tracked very extensively by Seabrook Station since the 70s as part of their NRC relicensing program. And you can see the pattern of clams populations, which are the red bars over this period of time. And then this yellow bar is the license sales for clam harvesting. And there's been always strikes me about this graph is the boom and bust pattern of the clams, going from high points to low points, and also the coincidence with the harvest pressure as indicated by the license sales, which if you talk to a biologist is following pretty much a predator-prey relationship. The other issue, though, affecting clams is green crabs, invasive green crabs, which prey on the juvenile clams. So there's a number of reasons that the clam populations have been held in lower numbers in most recent years. River herring is one of the species we track in terms of migratory fish, and it's tracked through returns to the fish ladders around Great Bay and Hampton Harbor. So basically every spring, fisheries biologists are going out to these ladders, and as the fish are trying to come up the ladder, they are having to be basically spooned over into the upstream areas and counted while that happens. But the pattern, this is a combination of all the returns to all the rivers in the Great Bay Estuary. You can see a general increase from the 70s up to 2000. That had a lot to do with the installation of the fish ladders, and then a sort of stabilization of the population. Since then, we have seen a bit of decline. We are not sure what the reason for that. It may have something to do with habitat. It may have something to do with the performance of the fish ladders. So in terms of response indicators, again, this is what we are doing now, we as people are doing to try and improve the situation. One of our main actions has been conservation. And there's been actually a very positive outcome with this. During this period, we've seen a large increase in the amount of lands conserved. In the last 3 years, we've been conserving about 7,000 acres per year, which is a good ratio when you think that we're adding almost 2,000 acres of imperviousness per year. I always kind of compare those ratios, the conservation land increase to the impervious surface increase. And this graph here on the right is showing where the conservation lands are more highly concentrated. You can see a large portion here around Great Bay having a lot to do with the Great Bay National Estuarine Research Reserve and the partnership therein. In Nottingham, where Piscataqua is, and up in the north in Moose Mountains. While we are conserving land on an aggregate basis at a good rate, we're not necessarily conserving it in places we need to. There was a very large planning effort done to identify conservation focus areas. I don't know what to do with this.
So don't knock it down.
No one does.
Very good.
So anyway, conservation focus areas. Areas that were selected by places like Nature Conservancy in New Hampshire and Maine of where high priorities to protect. Perhaps set a goal to try and conserve 75% of those by 2025. And unfortunately, we're only have 28% of those that are conserved. Making good progress in some areas, but we clearly are hoping to focus more effort into the high priority focus areas. And for restoration, there's 3 issues that we track. Oyster restoration, which is shown up here at the top, where we've had a slow and steady increase thanks to the efforts of the Nature Conservancy and Ray Grizzle at Jackson Lab. We now have restored 12 acres of oyster beds. And when you think that those are at the tune of $50,000 per acre, this is a significant investment of in our green infrastructure. Salt marsh in the top right. This was a restoration that was hot and heavy in the 2000s, largely around tidal restriction removal, trying to restore natural hydrology to the salt marsh. And we've set a goal to, now that we've removed most of the restrictions that can be removed, just start enhancing it, enhancing salt marsh. And we show this here in yellow, which is very hard to see. We're making slow progress on that, but we're starting to deal with that issue, like removing invasive species and improving habitats. Eelgrass restoration in the bottom right. Eelgrass restoration has been a real challenge. It's in-water restoration, so the quality of the water matters in terms of whether the plants can survive. And with the water quality having some problems in terms of eelgrass survival, plantings of eelgrass have not been particularly successful. Finally, this graph here on the bottom left shows our progress towards opening up our main stems of our rivers to migratory fish. And overall, we've opened up about 48 miles, which is about 50% of the total main stem miles and pretty close to our initial goal. So we are making good progress on some of our restoration, and some of it is challenged by the environment in which we do the restoration. So, in summary, estuaries are very challenging places, and I'm sure you all know that. They're some of the most complicated places on Earth. And I find just getting to this point, where you're summarizing from the million data points down to those few slides, a real challenge. And then I always find the summary slide to be the ultimate challenge. Because what are you going to say? What do all these different indicators say about the state of our estuaries? How do you put these together? How do you weigh different indicators? Well, the process we used was to work with our Science and Technical Advisory Committee to try and classify each of these indicators into a category. Whether it's a positive trend, which is shown in blue, whether it's a cautionary trend shown in yellow, or whether it's a negative trend shown in red. We put our restoration indicators down here in this green box because we're comparing those to our goals. It's just a goal that we're setting. It's an arbitrary goal. It didn't feel like it's an appropriate category to say whether the indicator estuary is getting better or worse. So, but of these 22 indicators, 15 are classified as negative or cautionary, and 7 are in the positive category. So overall, the assessment is that there's reason to be concerned about our estuaries, and that increased efforts to study them are warranted. The indicators show that we continue to face some serious challenges around the pressures on our estuaries related to impervious surfaces, nutrient loads, bacteria. They also show a positive thing, and that I can point to some of our indicators in restoration, that we have been able to move the needle and move in a positive direction when we put our effort towards it. When we invest in conservation and we invest in restoration, we have had a positive change. It hasn't been a lost investment. And so we hope to continue that effort over the next few years. And then when we start in 2016 to compile the 1 million+ data points again to make the new state of the estuaries, we hope that we'll be able to see the shape of this diagram change. See more things in the blue, less things in the red. I want to talk just a few minutes now about our current research and how we can link up with CCOM. There's a few things we're working on. This past summer, we did a pretty intensive aerial survey mapping of the Great Bay Estuary and actually the whole New Hampshire coast. This was done through ostensibly to help us with eelgrass mapping. It's 1-foot imagery that we collected at a spring low tide in August. Yes, there's a lot of imagery out there, aerial photography, but most of it's in April. Leaf off, collected by DOT, doesn't really have anything to do with the biology of the estuary. So it was really important for us to go out during the peak of the growing season at a spring low tide and have excellent water clarity. So we hope to be able to map not just eelgrass, but also oysters, maybe some other underwater habitats. And there may be a connection to some of the work that you do. We're also involved with a joint program with Long Island Sound to study the impacts of oyster aquaculture on nutrients in the estuary. There's been this question raised of, should we just grow more oysters in the estuary and use that as a way to extract nitrogen? And it's a good question. It has to be studied in a careful way to understand what is the true build-out potential for oyster aquaculture in our estuary, given the other constraints like mooring fields, areas that are closed to harvest, the research reserve. So that project is moving forward, and we should have a final report sometime this winter. And then finally, we've been working with Dave Burdick and Elizabeth Cianciola at Jackson Lab to do some more quantitative monitoring of macroalgae. As you know, a 2-point graph is not what we want to be seeing, so we want to add some more points to that graph to understand this pattern better. 2014, we were going to be forming partnership with Miracuz to do an analysis of all the data that's collected at the Great Bay Coastal Buoy. This is a large buoy in the middle of Great Bay. It's mainly been used for real-time reporting. This is just a snapshot of the data portal. The data itself has never really been compiled and analyzed beyond that. It's a fairly big effort, but we're going to gather a number of partners, get through the quality assurance issues, and then ask some questions and see what we can show with that data. The buoy's been out there for 7 years now. I mean, it feels like yesterday that it went out, but it has a lot more sensors on it than our existing buoys. And for instance, it has chlorophyll, CDOM, light attenuation, and other factors that we don't have normally that information for. So to wrap up, where can we interface best with CCOM? And in preparation of this, I was thinking about this and spoke with Derek to try and get some ideas. And here's what we're thinking. One of the most useful things for a lot of our work is bathymetry data. And that's something that CCOM does very well. And there certainly are bathymetry data sets for the Great Bay Estuary, but they aren't all stitched together in a way that we can use. And it would be really helpful if we could get all these data sets to be in one place or one datum that would help us to just use them as a base layer. We need this for 3 reasons. Things like eelgrass are depth dependent, and as we study this, where eelgrass can and can't grow, it's important for us to know clearly what the depth is, and we'd like to have a good base layer for that. It'd be really helpful, in particular, to have the seamless bathymetric topographic map so that we can model sea level rise better. I know there's a lot of work going on with that, but we'd like to get that as clear as possible. And finally, the thing you may not have thought of is sedimentation. there's been a lot of work to try and understand what's happening with sediment loads to the bay, where we have accumulation, where we have loss of sediments. And it's not an easy thing to measure, and it's very spatially dependent. But if we had repeated bathymetric surveys of the bay, we could do comparisons of these different bathymetric maps to look for areas of accumulation and for loss. The other thing that we thought of is that the work that we do to map habitats, such as eelgrass and oysters, is very intensive work. It's very site-specific work. And it would be really helpful if we could automate that a bit. And I know there's research going on about using acoustic methods to take the backscatter, do something with it, and I'm not going to even attempt to say the jargon because I don't know it, in order to understand what's happening in terms of your benthic habitat. And that would be certainly an area of research that would be helpful to us. So anyway, thank you all for your attention. I'm glad I didn't try and do this in Spanish. I think that would have been really hard. And we really look forward to collaborating with you all, either sharing data sets, developing research proposals, developing grant proposals. I think there's a lot of intersection between our work and your work, and now that we're all part of the same School of Marine Sciences and Ocean Engineering, we should collaborate more. Thank you. Any questions?
Have you done any economic value studies to sort of relate your indicators and the outcomes there to economic value?
We've started down that road. I can't say that we've done it and that it's an area of interest for us as we go forward. So if you're moving in that direction as well, we'd like to work with you on it. We wanted to.
Hoping maybe you.
No, well, where we got with that is saying that we needed to start small, and so we started to look at oysters in particular as an ecosystem service that had a market value that we could use as the kernel, and then build from there, talking about the in-plan models for effect on jobs. But that's a much bigger job. And it was fortunate for us that this partnership came along with Long Island Sound, because with that partnership, we were able to hire economists to help us with that. So we're going to be talking about the economic value of the aquaculture. We're going to be talking about the avoided cost of the aquaculture in lieu of traditional infrastructure. And we're going to stop short of the secondary impacts, which is where you start modeling how those primary jobs affect other parts of the economy. So we will have something this winter that will deal with that small piece of the puzzle, but we know that we're going to try and expand from there once we have a little bit of experience.
The other thing that's going on is Trev, with a number of partners, is starting a project looking at sort of estuarine spatial planning, focused initially on Great Bay, but also Hampton-Seabrook. And part of that will be, we have a NOAA coastal management fellow that we brought in. And in year 2, she's just in year 1 of her fellowship, sort of compiling a lot of spatial data sets. In year 2, she'll actually be applying this an approach called INVEST, Integrated Ecosystem Services and Trade-offs Analysis kind of approach. And the whole methodology there is you figure out some different management scenarios that are high priorities for you, and then you try to explicitly provide the ecosystem services valuation and look at kind of who wins and loses and what are the trade-offs in economic terms between different management scenarios. So it has yet to be determined exactly what the focus of that project will be, but there'll be something happening next year.
Did you look at correlations between all those different indicators that you mentioned? For example, it occurred to me that some of them, if they're extremely strongly correlated, you might not need to measure all of them, but maybe more importantly, you would just expect logically some of them to be positively correlated and some to be negatively correlated. And if you could show that, it might be a good sanity check. For example, the ones that have to do with restoration, you would expect that if you saw those ones go up, some of them related to the, the condition indicators would go down.
Yeah. Yeah, we have done that to the extent we can. There's a lot of these work on different time scales. And for instance, the obvious one that we want to show correlations with there's between nutrient stress and eelgrass loss. And one of the challenges for us is just the different time scales at which we measure these things.
Or the water quality in eelgrass. I mean, you should see those.
You should. Unfortunately, you see probably a 5 to 10 year lag in the response, and that's part of our problem of doing a traditional correlation. You need to add a lag time to represent the difference between when the stress occurs and when the effect is readily observed. We do attempt to do that whenever we can. Some of these are widely divergent, like bacteria and toxic contaminants, and those would be very different. But to the extent we can, we are trying to link these all up as in a causal chain. You have a question too?
Well, basic question is, what is the major source of dissolved oxygen?
Well, I mean, dissolved oxygen is, at full saturation is whatever, about 10 milligram per liter, depending on the temperature. But then it declines based on the algae growth and respiration, and also sediment oxygen demand, depending on where you are.
I understand what's causing the decline, and I'm not sure where it's coming from. Is it coming from aeration or?
Oh yeah. It's just the equilibration between the atmosphere and the water.
Right. Okay.
So that's when it's at full saturation, it's fully equilibrated.
But it's not like related to the power plant discharge from heating and cooling systems.
No.
I don't know if you mentioned this, I might have missed it, but did you ever look at the sensitivity to the Great Bay ecosystem to temperature changes, either in the water or the atmosphere? Long-term trends, maybe not so much short-term?
Yeah, we don't go back quite far enough to really be able to detect a trend in temperature, but it's a good question. It's one that comes up for us about things
What
like.
do you expect for the next 10 or 15, 20 years or longer?
Yeah, what would be whether that heat stress might be an effect on some of the habitats. There was a good study that Jen Dijkstra did at the mouth of the harbor, where she was looking at a temperature shift that as it related to invasive species colonization. And that was really a pretty isolated study where she could focus in and control a lot of the variables. So it was something that was a little bit easier to do than this full-scale analysis that we do.
The percent of shellfish beds that are permanently closed seems high, and I'm wondering, are these closed because the bacterial levels surrounding them are in the shellfish is always too high, or are these beds that are close to potential sources of pollution, say sewage outfalls, that are so close that they couldn't get the word out of contamination should there be a runoff of them or something like that?
Yeah, I mean, what you just described is actually a very good example, is exactly what the FDA makes us do. There's a lot of areas that are closed because the water quality just isn't good. But there are also a large area that are closed around wastewater treatment plants for exactly that reason. And they do a time of travel study, and they determine basically what is the area that will be reached by a plume that if the disinfection system breaks. And I think they use 4 hours as the cutoff to say it's going to take them 4 hours to notice, 4 hours to notify, 4 hours to do these other things. And so that area then is cordoned off as prevented for harvesting, even though the water quality may be okay in it. So it is a combination of those two things. And it isn't always just bacteria. Like, for instance, areas around the shipyard are closed due to potentially toxic sources. Most of the Piscataqua River, just because of the industry along it, are closed because of the potential sources. Yep.
You mentioned the clam populations in the Seabrook Harbor.
Yeah.
Is the main decline a factor of the green crabs?
Well, we haven't sorted that out exactly. That was one of the hypotheses. And we did a study where they actually put out huge nets on the clam flats to keep the great crabs out and to see what would happen. And there was a big improvement in the clam populations.
Do you think it might be beneficial to encourage predation on the green crabs to
Like
help the
human
crab?
predation?
Yeah.
We've been looking for a recipe for green crabs for a while, and they're just not all that appealing is the problem. But yes, obviously that's the issue, or that is one of the issues.
I know they're eaten widely in Europe, so.
Well,
it
if
might
you're
be
there,
interesting
find
to
out what the
try to
dish
encourage
is or
them.
how they're prepared or something, and you'll have to try that out here.
Green crab removal is very, very difficult.
The actual fishing of it?
Yeah, the actual fishing of it, but also just removal. Freeport has they've been doing studies for the past 3 years on green crab removal, and they've spent thousands of dollars, and it hasn't helped at all.
All right, well, thank you all for coming, and it's Friday afternoon. I'll be headed to Guatemala in less than 12 hours. This is the last of my English for a while, so I appreciate your attention and have a nice weekend.